Yeah, thanks a lot for this invitation and for giving me the opportunity to present with these great other people. And my project is on disentangling large-scale disease association data to enhance the reliability of AI-driven drug repurposing and drug target individuation in precision medicine. And so that's the background and motivation for the project. So one thing we're facing at the moment is that our current treatment for many complex diseases are actually pretty ineffective. So what you see here in red and green are fractions of patients that received a specific treatment and then either did or did not benefit from it. And in green, you see the fractions of patients that did, and in red, those that didn't benefit. And you see that actually for the diseases shown here, the situation is rather bleak, and this actually holds for many, many complex diseases. So now the question is, what is underlying this rather bleak situation? And there, one reason is that today we actually often treat rather umbrella diseases or phenotypes instead of diseases that are defined and understood at a molecular level. So take, for example, asthma as an example. So that's a disease which has historically been defined in terms of symptoms, but those symptoms are most likely caused by various possibly disjoint molecular mechanisms. And now if we treat all the patients that, well, in reality suffer from disjoint molecular disorders with the same treatment, and of course, we don't get very effective treatments. And now the same holds for many, many other diseases. So here we have hypertension as another example on that slide, but you could say the same for Alzheimer's disease, for Parkinson's disease, and for actually most so-called complex diseases. Now, the dream or vision of precision medicine is that we try to disentangle those umbrella diseases into their mechanistic subtypes. And then once we have understood those mechanistic subtypes, we can actually intervene in a much more targeted fashion and maybe come up with some disease-modifying treatments instead of just targeting the symptoms. And maybe sometimes it might even be sufficient to induce some lifestyle changes. So that's where we would like to go. So that's the long-term vision we would like to achieve. And now the question is, of course, how do we get there? And here, so sort of the classical thing to do would be to do classical biomedical research. So characterize a well-defined patient cohort in terms of molecular characteristics, phenotype data, maybe also do some animal model studies. And then based on those results, which you get from those types of studies, try to subtype your umbrella diseases such as asthma or hypertension into molecular subtypes. So that's a classical way to do. But now with the advent of AI, it's actually become increasingly popular to use a data-centric approach. And what's often done there is that you start with disease association data, which is available on the web. So there are, for example, databases such as DisGenet that contain information about disease gene associations, and then there are other databases where you have disease drug associations, disease symptom associations, and so on. And now once you have this data, you can download this data, and then you can take those associations to start your AI-driven analysis to find the molecular subtypes that cause your diseases. So, for example, what's often done is that you take disease genes from a database such as DisGenet, and then you could maybe project them onto some protein-protein interaction network and try to find interacting proteins that are associated with your disease. All right. So this is what we would like to do in a data-centric precision medicine. We take large-scale disease association data, we extract the disease associations we're interested in, and then based on those, we use AI tools to try to find mechanistic subtypes and in those individuate possible drug targets. That's the objective, but now there is a problem. And this problem consists in the fact that in those databases here, well, so there, in order to annotate the diseases for which we have the association data, we actually, of course, use the current phenotype-based umbrella disease definitions. Yeah? So we do not know the mechanistic subtypes of asthma yet. And because of that, in those databases, we of course, cannot annotate our diseases with the mechanistic subtypes. Yeah, but if we now use all the disease genes which are associated with such an umbrella disease, and now we try to find sort of, disease mechanisms based on those, we of course sort of, have all the blurriness which is in those definitions ingrained into our analysis. So that's a problem. And now, so you can now ask the question, to which extent sort of this annotation bias undermines this objective of data-centric precision medicine. And asking this question is exactly what we did in this recent publication here. So there we essentially try to answer the question how bad this problem is and whether those biases which are introduced by the annotations even out or not. And what we did in order to answer this question is the following. So we took several types of disease association data. So, for example, disease gene associations, disease drug associations, disease symptoms associations, and so on. And then we constructed disease networks based on those, such that we have diseases, so for example here, disease 6 and disease 1, connected by an edge if they share an association. So then you get different type of disease networks. So for example, in a gene-based disease networks, we sometimes call them diseasomes then, you would have an edge between two diseases if they share an associated gene. In a drug-based diseasome, you would have an edge between two diseases if they share a drug indication. All right, and now what we said is, okay, let's assume that the biases do even out and that those disease association databases, despite this annotation bias, somehow still contain useful information about disease mechanisms. So under this assumption, we can then say, well, in this case, then our disease networks should actually be systematically similar to each other. Yeah, because although the mechanistic subtypes are maybe not known yet, they should induce sort of parallel associations between the different data modalities. And this is now an assumption you can test. And that's exactly what we did in our paper. So we tested this assumption both globally for the entire networks and locally for the individual disease neighborhoods in those networks. And now what we actually found out is that for most diseases, those local neighborhoods are actually not systematically more similar than expected by chance. So what does this mean? So this means that if we take large-scale disease association data and we try to find disease mechanisms based on those, then we actually only get a blurred view at the disease mechanisms because the annotation bias prevents us from going more into detail. And that's, of course, a rather negative conclusion. And now the question is, what do we learn from that? So how do we deal with this outcome? Now, well, there's the sort of straightforward consequence, and the straightforward consequence would be to say, okay, well, let's just limit the use of large-scale disease association data in data-centric precision medicine, yeah, or just say, well, this only allows a blurred picture of disease mechanisms, and we might have to resort to classical biomedical research. So this is, well, this is one possible consequence, but of course, to say, let's not use that data, it's not so nice because this data, well, it contains sort of, the accumulated and aggregated output of a lot of research studies, and it would somehow be a pity to not make use of that. And that's now what I would like to propose here. So for this project, I would like to propose an approach where we try to disentangle the existing disease association databases in order to retrieve the unknown subtypes of our diseases. And for that, I will use a strategy which is actually often used in machine learning. And this strategy sort of says that if you can quantify a problem, then often you can use similar techniques which are used for quantification to formulate an optimization problem where this problem is addressed. And so in order to show how we do this, I have to go a bit more into detail into what we did for the quantification of the problem. So that's essentially sort of the method underlying the plots which I had on the previous slide. And so what we did is we took our diseases, and then for our disease D here, we took the associated genes for disease gene association, and then we looked at the other diseases which are also associated with those genes associated to D, and these other genes here are then the gene-based neighborhood of our disease D. Now you can do this for disease gene associations, you can do it for disease drug and disease symptoms associations and whatnot. And so, for example, for drugs, then you would also get such a neighborhood, now a drug-based neighborhood of our disease D. And now you can compare these two neighborhoods. And that's exactly what we did using some suitable distance measure. And now what we observed is that those distances are now, for most diseases, those distances are not significantly smaller than expected by chance. Now, that's the quantification, the problem. And now if we want to solve the problem, we just turn this around. And now we say, okay, so now what we would like to do is to split our diseases D and their associations into subtypes such that after the split, the resulting neighborhood distances are actually significantly smaller than expected by chance. So this is how this could look like. So before we had just one disease D here, and now after such a disentanglement, we would maybe have two diseases, D1 star and D2 star, and those then have two separate gene-based neighborhoods. And now we can do the same for the drugs, and then we get two separate drug-based neighborhoods. And now we can compare those. And of course, we do not only do this for our one disease D here, but we do this for all diseases in our diseasome simultaneously. And what we want to achieve is at the end is that the overall neighborhood distances, which we get in the entire networks, are now after sort of this disentanglement, are then significantly smaller than expected by chance. So the situation which we did not have with the original data, we would like to enforce that after this disentanglement. This is now a task which you can phrase as an optimization problem. So here, this is just a very high-level formulation of that. So we try to aggregate somehow the distances which you get after the disentanglement, and then we have to regularize the disentanglement such that we do not just end up with very small subtypes. And actually, all the ingredients here are still up to be defined. Yeah, so this is nothing which I have already fully conceptualized. We have to think about how to do this aggregation and how to do this regularization. How do we implement this?
Can you please?
One more slide. Yeah. How do we implement this? First of all, we need the association data, and for that we use existing tools like NetRex and BioCipher, and then we will solve this problem using local search, genetic programming, or integer linear programming. Last slide, output. How do we feed that back to the community? We will develop a database. You can run this database with queries such as return genes associated with your complex disease of interest, like asthma. So then this database gives you a set of genes for the subtype 1 and the subtype 2. We will provide functional characterization of those subtypes. For example, one would be then high inflammasome activity, and maybe the subtype 2 is characterized by enrichment of some immunological pathways. And now you as a researcher can say, okay, I'm interested in this asthma subtype, which is associated with immunological pathways, and I will only use those genes for then downstream AI-driven drug target individuation and drug repurposing. And that's it. With this, I would, that's almost it. So endpoints, how are we going to provide this? We will provide a web interface, we will provide a REST API, and we will provide a Python package in order to query this database. I would like to thank especially the co-authors of our Nature Comms paper, especially Hasepi, who was first author, also my lab, and our funders. And with that, I'm happy to take your questions.